Hello everyone, and welcome to episode 6, Remastered. Today, I'll be discussing the lipid class of biomolecules, and 3 of its major subgroups: fats, steroids, and phospholipids. Now you probably know that the fat in our bodies, and the butter that we put on our toast, are lipids. But did you also know that beeswax, and the wax on the leaf of a plant, are also lipids? Cholesterol and olive oil are lipids as well. In this episode, I'll cover the structure of lipids, their qualities, their functions, and the role that they play within cells. I'll spend a portion of the episode talking about membranes, which are a naturally forming structure composed of lipid molecules. And I'll talk about this because the membrane is an extremely important aspect of the cell, and life in general. The membrane is what defines the edges of the cell body. It regulates the in- and outward flow of ions and various molecules. It separates and protects the cell's internal chemistry from the exterior environment. Membranes allow the cell to concentrate various chemicals where they can be quarantined or easily accessed. And all of these things are critically important for the proper, stable functioning of living cells. Recall in episode 2, The Chemicals of Life, where I discussed hydrophobic and hydrophilic molecules. To briefly recap, remember that hydrophilic molecules are those that have polar or charged atoms, like an oxygen in a hydroxyl group, or a nitrogen in an amino group. These molecules can interact with the polar water molecules, and they're thus soluble in water. Hydrophobic molecules, on the other hand, have few, if any, of these polar atoms, and so they don't interact very well with water. When you put hydrophobic molecules in an aqueous solution, they tend to clump together so as to minimize the surface area that's exposed to the polar water. Due to their structure, lipids are overwhelmingly hydrophobic. They can't be dissolved in water or other polar solutions. The lipid class itself is very broad. It's defined as any carbon-containing compound that's both hydrophobic and nonpolar. These can be small chunks of carbon like isoprenes, free-floating chains of carbon like fatty acids, groups of chains like a triglyceride, or bulky heaps of carbon rings like steroids. Unlike proteins, nucleic acids, and carbohydrates, lipid molecules do not share a particular base of subunits or monomers that bind together to make polymers. Lipid structure is actually really variable. Chain length, saturation, ring formation, all of these things influence the structure and the function of the lipid. A simple fatty acid is a chain of carbon atoms with a carboxyl group at the end. A carboxyl group has a carbon atom with a double bond to an oxygen and a single bond to another oxygen that belongs to a hydroxyl group. The carboxyl group has the chemical formula COOH, which can be spoken out loud as CHO or CHEW for easy reference. These fatty acid chains with their carboxyl ends can clump together to form various glycerides like diglyceride or triglyceride. They can also bond to phosphate groups to become phospholipids, which are the primary molecules composing the plasma membrane. When carbon atoms form chains connected by single bonds, the carbon atoms can rotate freely, which makes three-dimensional bends in the chain. It's kind of like having a string of yarn that you're just shaking around. It's going to flop all over the place. However, when two of these carbon atoms, two adjacent carbon atoms, share a double bond, the carbon atoms here lose this freely rotating characteristic. If the double bond exists somewhere in the chain, it will produce a big kink in the structure. Double bonds do not allow their atoms to rotate around each other like single bonds do, so a double bond forces that region of the carbon structure into a single plane. Usually, fatty acid chains will form linear molecules, but double bonds can bend them out of shape. Sometimes, a chain will come to a double bond, and then it'll be bounced back, so to speak. This configuration, where two groups branch off of the same side of the double bond, is called the Syst Configuration. The alternative configuration, where two groups branch off of different sides of a double bond, is called the Trans Configuration. Trans configurations create roughly linear fatty acid chains, while cis configurations create an elbow bend, or an L shape in the fatty acid chain. When a fatty acid chain has no double bonds between any of its carbons, the chain can be called saturated, it's saturated by hydrogen. Every carbon atom in the chain, with the exception of the carboxyl carbon, have all of their free valence electrons shared with hydrogen atoms. Basically, the saturated fatty acid chain becomes a polymer with CH2 monomers. Now I know I said lipids aren't really polymers, because they're really not, but when you're talking in this special case about a saturated fatty acid chain, the monomers aren't big molecular chunks of stuff like a nucleotide or an amino acid or a glucose sugar or whatever. In this case, the monomers are just little CH2 units, a carbon atom with two hydrogens, and just that repeating. Now, fatty acid chains that do have one or more double bonds are called unsaturated fatty acids, because the chain isn't saturated with hydrogen. If it were, then hydrogen atoms would come and take the valence electrons from each carbon participating in the double bond. They would break up the double bond, and the chain would thus be saturated. When you have fatty acid chains with many double bonds, these are described as polyunsaturated. The saturation of a fatty acid molecule is really important to the macroscopic structure of the lipid and the role that it plays in the cell's physiology. First of all, the length of the carbon chain is really important. Short carbon chains don't really offer much in the way of structural integrity. It's kind of like trying to make a blanket by tying little tiny pieces of string together. Longer carbon chains, on the other hand, can interact in a way that's really somewhat similar to carbohydrates. You have long chains of fatty acids, and these can overlap or pile together to form large sheets, which can be stiff and durable. Waxes, like beeswax, are composed of saturated lipid molecules with long carbon chains. As a result, waxes like candle wax or the honeycombs of beeswax are stiff and solid at room temperature. Because of their linear structure, saturated carbon chains are really easy to stack together tightly. This inherent ability to be packed densely contributes to the stiffness that's characteristic of waxes. Unsaturated and polyunsaturated fatty acids cannot pack together as tightly as the saturated fatty acids can. This is because the kink in an unsaturated carbon chain is an irregularity that prevents the molecules from lining up cleanly side by side and crowding together, like they do in waxes. Because of this much looser organization between the lipid molecules, unsaturated fatty acids are usually liquids at room temperature. Olive oil, safflower oil, and really any other kind of organic oil substance are all composed of these unsaturated and polyunsaturated fatty acid chains. Now, all of these oils and these fatty acid chains are part of a subgroup of lipids called fats. Fats are composed of molecular structures called triglycerides, which are themselves composed of three fatty acid chains all bound together with a glycerol molecule. The glycerol molecule itself is relatively small. It's just a 3-carbon chain with each carbon bound to a hydroxyl group. This makes the glycerol molecule polar, reactive, and soluble in water. A condensation reaction will turn the oxygen in each hydroxyl group into an ester as it binds with the carboxyl group of the fatty acid chain. And then you have steroids, which are heavier lipids characterized by a 4-ring structure. Steroids are differentiated by whatever kinds of side groups that they happen to have attached to these backbone rings. Cholesterol is a very well-known steroid, and one of cholesterol's identifying features is a medium-sized carbon chain attached to the pentane ring. Phospholipids are similar to triglycerides, except one of the hydroxyl groups on the glycerol binds with a phosphate group instead of a third fatty acid. The phosphate group is polar. It carries a strong negative charge. The nonpolar fatty acid chains are repulsed by this polarity, and this pushes the fatty acid chains in the phosphate group to opposite sides of the glycerol. This also means that the phospholipid has 2 distinct parts: the hydrophobic fatty acid chains and the hydrophilic phosphate group. This dual nature makes the phospholipid an amphipathic molecule, or a molecule with both hydrophilic and hydrophobic regions. The amphipathic quality of phospholipids is extremely important, more so than any other quality that a lipid has. When placed in a water solution, amphipathic compounds don't really dissolve. Instead, the hydrophilic heads of the phospholipid interact with water, while the hydrophobic fatty acid tails crowd together to avoid water. Because of this, if you dump a group of phospholipids into a water solution, it'll spontaneously form micelles. A micelle is like a really small bubble. The outer surface of the bubble is a layer of hydrophilic phospholipid heads. The hydrophobic tails all hang inwards from the surface of the micelle, with the chains interacting with each other inside of the little bubble. This micelle structure allows the hydrophilic regions to interact with water, while protecting the hydrophobic regions from coming into contact with water. Micelles are really small. There's very little space, if any, left inside the bubble that isn't filled with fatty acid chains. But when collections of amphipathic molecules get large enough, they spontaneously form a structure that's slightly more complex than a micelle. They form a phospholipid bilayer. Each dot is the hydrophilic head of the phospholipid, with the fatty acid tails hanging down, going about half the distance through the bilayer. In this way, the hydrophilic surface of the bilayer protects the interior hydrophobic regions from water. The bilayer protects its edges from water by wrapping itself into a sphere or a bubble shape, with water both inside and outside the bubble membrane. Both micelles and bilayer membranes require no energy to form. They're simply the most stable resting states that large numbers of amphipathic particles take when they're dumped into a polar solution like water. The first cells that ever existed used these bilayer membranes to isolate and protect their internal biochemistry from the outside world. And this quality is so important that this trait has persisted in all life that's descended from this. Bilayer membranes give cells a very surprising number of qualities and abilities that life depends on. Membranes provide a degree of permeability. They can shrink, expand, they can bud little small bubble polyps out, and they can host proteins, which are involved in all kinds of cellular activities. Membranes can also be used to create concentration gradients that can be exploited for energy, or to compartmentalize caustic chemical reactions where they don't hurt other parts of the cell. The permeability of the bilayer membrane refers to its capacity for allowing certain particles, like ions or small molecules, to pass through it. Consider two aqueous solutions separated by a bilayer membrane. A solute is put into one of the two solutions. If the membrane is permeable to that solute, then the solute will begin to appear in the other solution, in the solution that it wasn't originally poured into. The rate at which this solute enters the second solution can be used to calculate precisely how permeable the membrane is to that particular solute. This phenomenon, where some solutes pass through the membrane easier than others, is called selective permeability. The bilayer membrane is selectively permeable to small, nonpolar, or weakly charged molecules, like CO2, molecular oxygen, water, that kind of stuff. These small particles can slide easily between the phospholipids. Larger molecules, or molecules that have a strong polarity or a strong charge, like ions, cannot easily move through the membrane without the assistance of specific proteins. However, not all membranes have the same permeability. Van der Waals forces are weakly polar interactions between atoms, created by the small polarity differences induced by the electron moving around the atom. The electron carries a negative charge, while the opposite side of the nucleus from wherever the electron is has a positive charge, or positive polarity, I should say. Membranes that are composed of phospholipids with unsaturated fatty acid tails cannot group together that tightly, and so they aren't getting close together enough to interact very strongly through van der Waals forces, and this reduces the total cohesion and strength of the membrane. And in contrast, it increases the permeability of the membrane. It's kind of full of holes. Conversely, recall how waxes, like beeswax, are given stiffness and durability by their long saturated fatty acid chains. Longer fatty acid chains can engage in more van der Waals interactions with each other, and this allows them to be packed tighter together. Membranes that are composed of lipids with long fatty acid chains are thus less permeable to solutes. Cholesterols can also be part of a membrane. Because of their large, clumpy structure, the bulky rings of a steroid, like cholesterol, will push into the membrane. It'll push out all of these phospholipids and it'll make them crowd together, and that makes them packed a little more densely. So if you consolidate all of this data, if you have a membrane that has short, unsaturated fatty acid chains with kinks in it, it's going to be far more permeable than a membrane that's studded with cholesterol and composed of long, saturated fatty acid chains that are tightly interacting with van der Waals interactions. You can see how all of these different structural qualities of the lipids come together to give the membrane these overall traits. But that's not all. Temperature also plays a role in the membrane's permeability. At room temperature, a phospholipid membrane is like a liquid in its consistency. The individual phospholipids can move laterally across the membrane, and at room temperature they usually move at about 2 micrometers a second. When the temperature rises, the movement speed of individual phospholipids, as well as the permeability of the membrane as a whole, will increase. When the temperature drops, the movement speed slows, and permeability decreases. If the temperature drops far enough, the membrane can freeze into a solid and lose all permeability. When you put a mass of particles into a solution, their random movement will cause them to slowly diffuse, or expand outward to fill the container, finding a level of equilibrium when the solution has a single, uniform concentration of the particle, and there's no more net movement in any direction. The diffusion of water particles is called osmosis, and it occurs in membranes that are selectively impermeable to most or all other solutes. Imagine two solutions separated by a membrane. What if one solution had a higher concentration of solutes than the other, but that solute could not pass through the membrane through diffusion to reach equilibrium? What would happen then? Well, there's still a concentration gradient at work here, but it isn't the solute's concentration gradient. It's water's. If you have two equal solutions, and you put a solute in one solution, that solution will have a higher concentration of the solute. But a little less obviously, it will also have a lower concentration of water. Osmosis is then the net movement of water molecules going from areas of low solute concentration to areas of high solute concentration, so as to dilute them and find equilibrium. Now this is cool and all, but what does it have to do with life? With cells? Well, recall that a bilayer membrane forms a bubble, or a vesicle, which I'll just call a cell, and it has water on both the inside and the outside. If water leaves the cell, it shrinks. If water comes into the cell, it swells. If the solution inside the cell is isotonic, then this means that it shares the same solute concentration as the outside fluid. There's no net movement of water through osmosis, and the cell retains its shape. Now assume that our cell has a very low concentration of solute within its membrane, which makes it hypotonic relative to the external solution. This is equivalent to saying that the cell has a very high internal concentration of water. Naturally, the water will leave the cell, and the cell will shrink. A membrane that's shrinking can look crumpled and wrinkled, and if the cell shrinks too much, it can collapse and become unable to be reinflated. Now assume the opposite case, where the cell has a very high concentration of the solute within its membrane, which makes it hypertonic relative to the external solution. Water from the exterior solution will come into the cell, so as to reduce its solute concentration and dilute it to reach equilibrium, and this inflow of water will cause the cell to swell. Cells that have a dangerously high solute concentration can swell so much that they burst, they explode, with their membranes ripping apart and collapsing like an overfilled water balloon. This generally kills the cell, as the membrane collapse destroys all of the carefully regulated concentration and chemical gradients and exposes all of the sensitive internal biomolecular machinery to the chaotic, dangerous, unregulated external environment. This capacity for membranes to swell or shrink plays an important role in numerous biological functions. Many plants, for example, have little pores in their external tissues called stoma, which are involved in the regulation of gas exchange. When the plant needs to open the stoma, the guard cells on either side of the pore become flooded with solutes, which makes their internal solutions hypertonic, and thus attracts water through osmosis. The guard cells swell as they fill with water, and since the tips of the guard cells are like stapled together, all of this swelling will force the cells to bow outwards, creating the opening of the pore between them. Gases can then flow into or out of the pore. Remember earlier when I mentioned the proteins that help move larger molecules and ions through a membrane? These proteins play a critically important role in the proper functioning of a cell's membrane, which is why I'm discussing them now and not in episode 3. Speaking of episode 3, remember how I discussed the side chains on the amino acids? Some of these side chains are nonpolar, while others are polar. When a contiguous sequence of amino acids has nonpolar side chains, and the polypeptide folds up to form a protein, that particular globular segment of the polypeptide is hydrophobic. Likewise, segments of the polypeptide with a large number of polar side chains are going to be hydrophilic. Now, just like the phospholipids, these proteins are amphipathic. When these proteins are placed into a membrane, the hydrophobic region is attracted to the other hydrophobic fatty acids, and here they can be stabilized by their own nonpolar interactions. The hydrophilic regions of the protein stick out on one or both sides of the membrane. Together, the various phospholipids and membrane proteins compose the cellular membrane in a kind of liquid mosaic. If you want to learn more about this, look up the fluid mosaic model, which is the current working model for the overall composition and organization of the membrane. So what are these proteins actually doing in the membrane? What are they doing here? Well, some of these proteins, like glycolipids, are involved in cell signaling and identification. Others are involved in physically stitching the membranes of different cells together to form tissues. But to stay on topic, I'm going to be discussing the membrane proteins that affect the membrane's permeability. Of these, there are 3 main groups, each with a wide variety of structures. These groups of proteins, in order of increasing complexity, are called channels, carriers, and pumps. Channel proteins are selective tube-like structures that allow a specific ion or molecule to move across the membrane. The interior lining of an ion channel is often charged or polarized to attract the specific ion that it shuttles across the membrane. Channels that move water molecules are called aquaporins, which literally means water pore. Then there are gated channels, which open and close in response to a particular stimulus. Voltage-gated channels, for example, only respond if the membrane is depolarized to a particular voltage. These voltage-gated channels exist in your neural cells. Ligand-gated channels only open if they bind to a specific ligand, like a sodium ion or a neurotransmitter or a hormone, or whatever it is that binds to their receptor. All of these channel proteins require no energy input to work. Carrier proteins, the second group, are very similar to channel proteins. They're tube-like proteins that provide a route for specific particles to cross the membrane, while also requiring no energy input. Carriers differentiate themselves from channels by the manner of their transport. Carriers are kind of like gated channels, except the item that they have to bind to open is the same molecule that they transport. A literal textbook example is the Glute transporter. All cells use glucose, either to store energy or for structural purposes. But unfortunately for cells, glucose is a pretty large molecule, and it's dotted with all these polar hydroxyl groups, so it has a really hard time diffusing across a membrane. The Glute transport protein is like a tube with its middle section squeezed closed. Outside of the cell, a glucose molecule is attracted to the Glute protein, where it binds to the protein's active site. Once bound, the Glute protein begins to bend and contort. Its shape will change in response to the bound glucose. So as the Glute transporter is altering its shape, the glucose molecule is being dragged through and released on the inside of the membrane. After it brings a particle into the cell, the Glute transport protein will let go, and naturally reverse its shape after letting go of the glucose molecule. In a more general sense, this is what all channels really do. They'll bind to something, change shape, release it, and then change back. In other cases, some of these carriers will bring their cargo in and let go of it, but then they won't do anything else. They'll have to bind to different ions inside the cell, change shape again, and then push those outside the cell. And then they'll need that first ligand to come and bind again to change back. The third group of these proteins are called protein pumps, and these are capable of moving ions or molecules against their concentration or electrochemical gradients. Unlike channels and carriers, moving particles against their concentration gradient or their electrochemical gradient or whatever, that takes energy. So in order to work, a protein pump has to consume ATP. The third phosphate group breaks off of the ATP and binds to the pump, giving it the potential energy that it needs to change shape, in addition to the polar interactions that it has with the protein side chains. Now, some protein pumps move hydrogen ions. Others move calcium ions. A really important pump in your neural cells moves sodium and potassium ions across the membrane. Protein pumps of all sorts can be used to maintain or to create electrochemical or concentration gradients, and these gradients can themselves be exploited as a source of energy. Co-transport occurs when one pump sets up a gradient, and this gradient provides the energy for one or more different pumps to function. Now, speaking of energy, I very briefly want to mention lipids' role in energy, as lipids are powerful energy storage molecules. In the same way as carbohydrates, the chemical energy stored in a lipid can be found in the bonds between non-electronegative atoms. Consider that carbohydrates have a carbon backbone where energy is stored, but they also have a lot of hydroxyl groups with electronegative oxygen atoms. With lipids, there are no hydroxyl groups lining the carbon chains. There's just non-electronegative hydrogen, and this increases the lipid's energy storage capacity tremendously. Fatty acid chains carried around on glycerol molecules are basically little packages of energy, just waiting to be broken apart and metabolized. Okay, so that was quite a bit about lipids, but I think I covered most of what I needed to. So to summarize this episode, lipids are largely hydrophobic, carbon-rich molecules. In aqueous solutions, some kinds of lipids can spontaneously form micelles and bilayer membranes. These bilayer membranes would naturally form bubbles, which were then worn by self-replicating molecular machinery like a cloak to make the first cells. Cells use these bilayer membranes to create isolated chemical environments. They isolate their internal chemistry from the outside environment, and they internally isolate caustic reactions in smaller little bubbles. The various membrane proteins allow the cells to maintain these isolated chemical environments, which in turn keeps the cell alive. And that is about it for lipids. This is also the final episode in this first series of the podcast. Now in the next series, I'll expand my focus and step away from biomolecules to start exploring the massive structures that they form, called cells. If that sounds interesting, then be sure to come by and check it out. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button and share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.